Cells are the building blocks of all living organisms. They perform all functions necessary for survival. Cells come in many forms like muscle, nerve, and blood cells. Each cell has a complex internal structure. The nucleus houses DNA, the blueprint of life. Cells contain organelles like mitochondria, ribosomes, and lysosomes. They are responsible for growth, energy production, and reproduction. Understanding cells helps scientists learn how organisms operate. DNA carries the genetic instructions for life. It's made of nucleotides, each with a sugar, phosphate, and nitrogenous base. The four bases are adenine, thymine, cytosine, and guanine. Their sequence forms a code read by the cell. DNA is organized into chromosomes. Humans have 23 pairs, inherited from each parent. Watson and Crick's discovery of the double helix was a milestone. Understanding DNA is key to new medical treatments. Cells read and execute DNA instructions to function properly. Transcription copies DNA into mRNA. mRNA is transported out of the nucleus. Ribosomes read mRNA in the cytoplasm. This is called translation. Ribosomes assemble amino acids into a polypeptide chain. Cells ensure correct protein production. Regulatory proteins enhance or inhibit these processes. Regulation maintains homeostasis. Errors can cause diseases. Cells have mechanisms to correct mistakes. Proteins are essential molecules that play diverse roles in the cell. They can be enzymes, which catalyze biochemical reactions, or structural proteins, which provide support and shape to the cell. Other proteins are involved in signaling, transport, and defense. The process of protein synthesis begins with the transcription of DNA into mRNA. The mRNA then travels to the ribosomes, where it is translated into a polypeptide chain. This chain undergoes folding and modifications to become a functional protein. Each step of protein synthesis is tightly regulated. Transcription factors control the rate of gene expression, while ribosomes ensure the accurate translation of mRNA. Chaperone proteins assist in the proper folding of newly synthesized proteins, and post-translational modifications can further refine protein function. The complexity of protein synthesis reflects the complexity of cellular functions. By producing a diverse array of proteins, cells can perform the myriad tasks necessary for life. Understanding this process is key to developing treatments for diseases caused by protein malfunction. Section 5. Cell differentiation, becoming specialized. Cell differentiation is the process by which a generic cell becomes specialized to perform a specific function. This is guided by the expression of different sets of genes within the cell's DNA. Differentiation allows for the development of various cell types, such as muscle cells, nerve cells, and blood cells, each with unique structures and roles. During development, stem cells differentiate into specialized cells through a series of regulated steps. This involves changes in gene expression, which are controlled by transcription factors and signaling molecules. The environment of the cell also plays a role in guiding differentiation. Differentiated cells have unique protein profiles that enable them to perform specialized functions. For example, muscle cells produce actin and myosin, proteins necessary for contraction, while nerve cells produce neurotransmitters for signal transmission. Understanding cell differentiation is important for regenerative medicine. By manipulating the differentiation process, scientists hope to develop therapies for replacing damaged tissues and treating degenerative diseases. Section 6. Cell division, replicating the code. Cell division is the process by which a parent cell divides into two daughter cells. There are two main types of cell division, mitosis and meiosis. Mitosis results in two genetically identical daughter cells, while meiosis produces gametes with half the genetic material. Before a cell divides, it must replicate its DNA. DNA replication involves multiple enzymes, such as DNA polymerase. During mitosis, chromosomes are evenly distributed to daughter cells. Understanding cell division is critical for cancer research. Section 7. Repair and maintenance, keeping the code intact. Cells have sophisticated repair mechanisms to maintain the integrity of their DNA. DNA can be damaged by various factors, including radiation, chemicals, and errors during replication. If left unrepaired, this damage can lead to mutations and diseases. One key repair mechanism is nucleotide excision repair, which removes damaged sections of DNA and replaces them with the correct nucleotides. Another is mismatch repair, which fixes errors that occur during DNA replication. Cells also have systems to detect and repair double-strand breaks, which are particularly dangerous. In addition to repair, cells have mechanisms to maintain the stability of their DNA. Telomeres, the protective caps at the ends of chromosomes, prevent the loss of genetic information during cell division. Telomerase, an enzyme, replenishes telomeres in certain cell types, such as stem cells and germ cells. Understanding DNA repair and maintenance is crucial for developing therapies for genetic diseases and for preventing cancer. By enhancing the cell's natural repair mechanisms, scientists hope to reduce the incidence of mutations and improve health outcomes. Section 8. Genetic mutations errors in the code. Mutations are changes in the DNA sequence that can occur naturally or be induced by environmental factors. While some mutations are harmless, others can disrupt normal cellular functions and lead to diseases such as cancer and genetic disorders. Mutations can occur in various ways, including point mutations, which are single nucleotide changes, insertions, deletions, and chromosomal rearrangements. The impact of a mutation depends on its location and the function of the affected gene. Cells have mechanisms to minimize the occurrence of mutations. During DNA replication, DNA polymerase has proofreading abilities to correct errors. Additionally, repair mechanisms fix damaged DNA. Despite these safeguards, some mutations escape detection and become permanent. Mutations play a key role in evolution by introducing genetic diversity. Beneficial mutations can be passed on to future generations, contributing to the adaptation of species. However, understanding and mitigating the harmful effects of mutations is essential for improving human health. Section 9. The role of DNA in evolution. DNA is the foundation of evolution as it carries the genetic information that is passed from one generation to the next. Variations in DNA sequences caused by mutations provide the raw material for evolutionary change. Natural selection acts on these variations, favoring traits that enhance survival and reproduction. Over time, beneficial mutations accumulate, leading to the development of new species and the adaptation of organisms to their environments. This process of evolution is driven by changes in DNA and is documented in the fossil record and genetic studies. The study of DNA has revolutionized our understanding of evolution. By comparing the genomes of different species, scientists can trace their evolutionary relationships and identify the genetic changes responsible for key adaptations. This has led to new insights into the history of life on Earth. Understanding the role of DNA in evolution also has practical applications. It can inform conservation efforts by identifying genetically distinct populations and help in the development of new agricultural and medical technologies. Section 10. Implications for science and medicine. The understanding of cellular programming and DNA has profound implications for science and medicine. By deciphering the genetic code, scientists can develop new treatments for diseases, such as gene therapy, which involves correcting defective genes to treat genetic disorders. Advances in genetics have also led to personalized medicine, where treatments are tailored to an individual's genetic makeup. This approach can improve the effectiveness of therapies and reduce side effects. For example, cancer treatments can be customized based on the genetic profile of a patient's tumor. The study of DNA has also enabled the development of biotechnologies, such as CRISPR, a tool for precise genetic editing. This has opened new possibilities for research and therapy, including the potential to cure inherited diseases and engineer crops with enhanced traits. In conclusion, the cellular programming language encoded in DNA is fundamental to life. It governs the development, function, and evolution of living organisms. Continued research in this field promises to unlock new scientific discoveries and medical advancements, improving the quality of life for future generations.
So hit that subscribe button, ring the bell for notifications, and let's turn the page on a new adventure together. Get ready to experience books like never before, only on VBook 360. Like, comment, and share to support the channel and keep the video magic alive. Thank you for stopping by. And remember, every video has a story, and every story deserves to be shared. See you in the next video.